Good. Today we're gonna discuss about a new topic that is called virology. It is part of microbiology that is studying viruses. And in the beginning, I would like to give you a question. Why computer viruses are called viruses, but not bacteria? Or fungi? Or protozoa? Or prions? Why computer viruses are called viruses? One by one, please. Oksina. What do you mean? What do you mean?
It acts just like how computer programs are.
They act like computer programs. Okay. So why they are called viruses?
Just like how we
Silence.
cells and produce in your
Please.
For the viruses, there are only RNA and they make our cells, humans body to work all the things. That's why the computer virus is also like that. They have a single programming and they go to your computer and use your data and system to filter their.
Very good. To both of you. So, indeed, if we make comparison between action of viruses in biology or in living organisms and action of viruses in computers, we'll observe some similar features and some similar actions. And if we start describing what virus is actually, talking about computers, any computer, it doesn't matter, is it PC or phone or tablet or any other device that has some operating system or something. You will observe that any computer is divided psychologically, or actually is divided in two main parts. Is hardware and software. When we are talking about hardware, it is everything we can touch, starting with the screen, keyboard, mouse, anything, and finishing with all the physical particles that are enclosed inside of computer. You can open it and you can touch it. Physical part of computer. And software. Software is something you cannot touch, but you know it exists, right? And software is, when we are talking about software, it is operating system, programs, data we have in computer. And software is made of what? Software is made of codes, binary code, 01, 01, 01. It is binary code for computers. When we are talking about any living cell, because whenever we are talking about viruses, it is mandatory intracellular, talking about microbiology. Viruses are mandatory intracellular parasites. But we also have some bacteria that are mandatory intracellular parasite. For example, rickettsia, chlamydia. They are also intracellular parasites, but they're not viruses. Which is the difference? Is the virus a living organism, living microorganism? Not really. It is a cellular unit that contains genetic material. That's it. Which is the difference between bacteria and virus? Bacteria is somehow independent microorganism. Of course, all of us as micro or macroorganisms in nature are dependent on something. But when we are talking about organisms, macro or micro, by calling them independent, we mean they have their own structure, their own morphology, they have their own physiology, their own genetical material, their own activity. They live, consume, produce, and multiply by themselves. Even bacteria that is mandatory intracellular parasite, for example rickettsia, that is able to multiply only inside of the host cell, they are having their own organism, their own structure, their own metabolism, and they multiply by themselves. When we are talking about viruses, the only thing that is common between viruses and microorganisms is presence of genetical material, either DNA, either RNA, just one of them, and presence of biological code that is called capsid. So some viruses are enclosed, or some genetical material of virus is closed in capsid, and some of them are closed in supercapsid. Supercapsid is an extra code that covers viruses. So they don't have their own mechanism for multiplication, their own organella that are responsible on biosynthesis, on processing of some microelements, of consumption and of production. They don't have any organella. They have just code and genetical material. So different viruses, as a result, will have affinity to different cells. That means not any virus can penetrate any cell. Not any virus can multiply in any cell. This is why whenever we are talking about viruses, we should understand that different viruses will affect different cells, tissues, plants, animals, humans, and as a result will produce different affections. How it happens? A virus while having its genetical material inside, in the moment it has a direct contact with a cell that can be affected by this virus, how this moment of connection happens. Viruses can have some proteins, some, let's say, keys, that are complementary to some cellular structures. So if the virus has this connection to a cellular, to this cellular structure, specifically to its cell membrane, that means this key will open the door. and connection between virus and cell will happen like the code of the virus, the capsid of the virus, will become part of the cell membrane, and by this connection it will open the inner part of the virus inside of the cell, and the genetical material will penetrate inside of the cell. What happens next? This genetical material will arrive into the cellular replicative organelle. Ribosomes, maybe it will come to nucleus. So all the processes of replication of genetical material, it depends, is it RNA or DNA, will start replication of this genetical material. This is how viruses are acting in computers. Just as your colleague told us, they act like other programs, right? So actually computer viruses are also a program. The computer virus is a program that after being installed in computer will change the destination of computer. So if before the function of computer was making all the commands of a user, of the owner, from this moment on, this computer thinks its destiny is making everything virus will command. So from this moment, the user, the owner, will lose totally or partially will lose control on his own computer. Same thing happens when virus penetrates in the cell. If till now the function of cell was production of some substances and its own replication, multiplication, because cells are able to divide, to multiply, from this moment on, the cell is hacked, is attacked by a virus, and the genetical structure of the cell will change. As a result, the program that is encoded in the cellular genetical material will be changed. As a result, the cell will think its function is reproduction of viruses. So actually the cell, the infected cell will become a factory of viruses. The virus will use all the facility of the cell it attacked to reproduce its own particles. As a result, cells will the infected cell will reproduce viruses until which moment? The cell will reproduce viruses until either the place in the cell will finish and it will explode and eliminate all the viruses outside, either some viruses are produced like this. The genetical material inside of the cell is replicated. It goes to the cell membrane. Cell membrane makes kind of budding. It closes the genetical material of virus and it goes out the cell. So the cell will reproduce viruses until the moment it will explode and it will release all the viruses outside, or until the moment all the cellular potential will be finished. So the cell will simply disappear. This is why there is a difference how viruses will act infected organisms. There is a difference between bacteria and viruses. How you make difference between bacterial infection and viral infection? If we are talking about bacterial infection, they don't penetrate inside of a cell. They are living near cells. They can produce something that will destroy cells of the body. The immune system will start producing and carrying in the inflammatory, in the place of inflammatory process, will bring a lot of phagocytes, and the fighting with bacteria will start, right? Phagocytes will try to swallow and to destroy bacteria. The inflammatory process will start, and this is how happens pus production. Because pus is, the main contents of pus is dead phagocytes with dead or not bacteria, and with rests of tissues of own cells. All together they will make this homogeneous substance, semi-liquid substance, we call pus. And if you will observe in big amounts, it will stink, right? Because everything that enters in contact with bacteria, pathogenic, conditionally pathogenic, non-pathogenic, the result of bacterial metabolism, it is the only thing in this nature, in our nature, that stinks. Did you ever think about it? And fungi also. Those saprophytic ones. Because they are responsible on decaying of biological substance, of organic substance. And the process of decaying, all the time there will participate either bacteria, either fungi, and it will produce a specific smell we call stinking, right? Now, when we are talking about viruses, first of all, phagocytes will be useless against viruses. For a simple reason, virus is not outside the cell, virus is inside the cell. And it is not that easy to detect virus in order to catch it, swallow and destroy. So it will appear inside of the cell, it will consume all the facilities of the cell, and after appearance of the virus inside of the cell, affected cells will start simply disappearing. So if you will see the structure of tissue, multicellular structure of a tissue, you will observe that tissue affected by viruses will have free spaces without cells. They simply disappear. They are like dissolved by viruses. And this is the only difference between biological viruses and computer viruses, because if the virus will damage the computer, the only thing it can damage is data. So finally you can make hard reset, you can format HDD, you can clean all data, everything. You will lose probably the data, but you can save the hardware, you can save the computer itself by reinstalling of the software, of the operating system and so on. But when we are talking about biological viruses that infect living cells, they will actually destroy the cell by its own multiplication. They will change the destiny of the cell and the cell will start simply reproducing viruses. Whenever we are talking about the morphology of viruses, as well as I told, two main parts of virus are code or capsid and genetical material, and different parts, different particles of the virus will represent different antigenic structure. This is why one of the mostly efficient ways to fight with viral infections will be which one? If they have their own antigenic structures, one of the mostly efficient ways of fighting with viruses will be administrating of what? Louder? Administrating of antibodies. Antigen, whenever you can hear antigen, you should think about antibodies. Whenever you think about antibodies, you think about antigen also. These two structures mostly all the time are meeting each other. So, on the surface of the virus, you will observe some similar structures. So you can see different types of viruses here, for example, and all of them, they are different in morphological structure, but still they have something similar. And one of the mostly similar things on the surface of the viral capsid is spike protein. I guess you heard about spike protein during pandemic and especially during production of the vaccine against COVID, right? Spike protein is essential, is very important because spike protein is actually that key that makes possible penetration of virus inside of the cell, and at the same time spike protein is that particle that makes possible binding of virus by antibodies. So it is a proteic structure that has function of binding, function of connection to surfaces or to cells in order to penetrate inside, and at the same time it can be catched, the virus can be catched specifically by these spike proteins. Whenever it were discussed production of antiviral vaccine for against COVID, against coronavirus infection, as well as you could remember, every distributor and producer of vaccines were explaining that look, you shouldn't be afraid of this vaccine because it doesn't contain information, genetical material of the virus itself. It is harmless. It doesn't contain information about virus. It contains just information about spike protein, about the key of the virus. So whenever this vaccine will enter the body, our immune system will catch this data and will introduce in its own database information about spike protein of this very virus. The immune response will happen, the data will be introduced in our database, and next time when actually the virus will penetrate inside of your body, your immune system will recognize it and will start producing antibodies against this spike protein. We don't need to fight with DNA of the virus. That's enough for us to fight with the key that opens our cells in order to let virus penetrate inside of the cell. So the vaccine was efficient and is efficient enough just to catch the virus and neutralize it, because if the antibodies produced against this spike protein will be produced in enough amount and will bind all the spikes of the virus will not have any way to attach to the cell. As a result, any living structure, even the virus that actually is not living microorganism, has its own life cycle, after which if it doesn't multiply, it will die. Or at least it will not multiply and will not produce, it will not enter the cell, it will not reproduce. As a result, it will not produce any harm to a host. So finally it will die because without producing any harm to a host. Another thing that makes viruses, what makes viruses being able to penetrate inside of the cell? What makes them to be able to penetrate inside of the cell? If you will pay attention to the mechanism, how they penetrate, how they multiply, and how they leave the cell, you will observe that virus is made of the own materials, of the cellular materials. Did you get it? This virus you can see here is made of the cellular materials. So it penetrates the cell, it is produced by this cell, and when it comes to the borders of the cell, the particle of the cell membrane will engulf the genetical material and will separate from the cell in form of bud. And this is the way of virus production. So how do you think, if the capsid is made of cell membrane, how easy for the virus is to attach and to penetrate and to become again part of the cell? Not too complicated. This is the way that makes viruses to attack again and again new cells, first. And second, this is how we explain specific affinity of specific viruses to specific cells. So some viruses will affect just some specific plants. Some viruses will affect just animals, and in those animals they will affect just some specific organs, some specific tissues, and will produce zoonotic infections. That means these viruses will affect only those animals. This is what used to be with COVID, with coronaviruses, right? Coronaviruses used to be zoonotic viruses. But due to some mutations, and here's the question remains open, with help of someone else or not, but after some mutations in the viral structure, they became able to penetrate in the human cell. As a result, they became zooanthropogenic infections and started multiplying in humans more than in animals. This is what also creates a lot of doubts. But still, the fact is fact. This virus mutated and started affecting people, started affecting humans, and specifically their specific parts, and especially respiratory tract. How can we detect viruses in the lab? Lab investigation of viruses. If we're talking about microscopy, I guess that's clear that the only microscopy we can apply to viruses is electron microscopy. This is why, please pay attention. Please wake up and pay attention. This is why when you have exam, when you have final on microbiology, and there is a picture on the screen, black and white picture on the screen. And here's the question, which is the pathogen on the picture? And few options, 5 options. Usually 4 of them are bacterial, and just 1 of them is viral. And white and black picture, whatever it is, 99% probability will be electron microscopy. Electron microscopy is all the time monochrome image. If you ever saw viruses in color, it is either painting, either post-reduction, post-processing of the picture. So electron microscopy is, do you know how it acts, electron microscopy? It's a device without any optics, like light microscope, without any lenses. It has a producer and receiver scanner that produces and receives the jet of electrons. It is not about electricity, it is about emission of electrons. Electrons are extremely small units. So when these electrons are penetrating the material we are investigating, and they are either reflected, either penetrating through this material and are received on matrix, they will give us the picture of everything they had contact with. So these small units, these small electrons, will give us the picture of everything they entered in contact with. As a result, electron microscopy is one of the mostly precise from morphological point of view techniques of investigation. And after we receive a digital image, monochrome, white and black image of virus, after that, just in order to make a little bit more evident, we can either draw it separately, either making post-reduction of the picture to make some spikes being red, some DNA to make blue, or something else. So whenever you can see monochrome picture of a pathogen, you already should understand it is a virus. It is a viral infection because it is made by electron microscope. It is a small tip. And by the way, one of the mostly easiest questions on the exam are based on pictures. And when I can see students misanswering questions with pictures, it is too evident if you know the basis. Except microscopy, but you should understand that microscopy is not applied in diagnostic in diagnostic in diagnosis. It's not applied in diagnosis. It's applied in scientific investigation. Simply because electron microscope is a very complex and expensive device, and the maintenance of the microscope is not cost efficient for any lab, for any clinical lab. It is efficient for scientific reasons, but not for diagnostic, not for clinical reasons. This is why one of the mostly efficient, fast, and useful methods of investigation will be molecular biological methods, which is the mostly spreaded one is PCR, polymerase chain reaction. Also, it can be RT-PCR, but please don't misuse. There are two types of RT-PCR. It can be reverse transcription PCR. It is applied for which viruses? For retroviruses. And RT-PCR also can mean real-time PCR. That is a little bit faster than simple PCR. So here you can see real-time PCR, and here is written qPCR or quick PCR. Yes? And RT-PCR, like reverse transcription PCR. It is used for converting of viral RNA into DNA. As a result, we can detect RNA viruses. And immunoassays. Immunological investigation. So whenever we are talking about immunological investigation, we are talking about, again, interaction between antigen and antibody. So all of these investigations will be divided in two main groups. Either serodiagnosis, that means detecting of unknown antibodies by using known antigen. Serodiagnosis is made to detect immunity, to detect presence of antibodies in the bloodstream. That shows either that a patient is already immune against this infection, maybe because of infection, maybe because of vaccination. And if we check types of antibodies in the bloodstream, it can be IgM, IgG. IgM will tell us about acute infection or about recent infection. IgG will tell us about chronic infection or about infection that passed long time ago. Whenever we are talking about seroidentification, that means we want to detect unknown antigens by using known antibodies. In this case, we take as a specimen not necessarily blood, but prelevates from the affected area. So if it is viremia, that means viruses in blood, we will take blood for investigation. But if it is a respiratory tract infection, we'll take the swab. Who did the swab during COVID? I guess everyone. And if you will pay attention, you did the swab for both PCR and antigen test. You didn't make a swab only for antibody test. If anybody made ever an antibody test, I'm pretty sure we have here some people who didn't make antibody test, maybe the majority even, but I'm pretty sure there is no person here who didn't make PCR test or antibody test, at least one of them, at least few times during the entire pandemic, right? Just because it was mandatory to leave the country, to enter the country, to while you've got sick, and so on. It was mandatory. Now, if you will get infected with coronavirus, nobody will propose you. Oh, still we make some, but almost nobody will propose you to make antigen test or PCR test. Just because it became as routine as flu or any other acute respiratory viral infection. But in that time, it was something normal. It was something very important to appreciate how many people got infected with COVID. So, these are three main directions of viral detection, just as we discussed. And one more thing about viruses, and please don't misuse macrophages with bacteriophages. Because when we are talking about macrophages and phagocytosis, about phagocytes, we are talking about actually engulfing of something by our immune cells. Because phagos in Greek means to engulf, to swallow. But whenever we are talking about bacteriophages, we are talking about viruses actually that are able to attack specific bacteria. So actually, in this case, not the virus will swallow the bacteria, but the bacteria will swallow the virus. But as a result, bacteria will be destroyed by virus. So bacteriophages are specific viruses that can attack specific bacteria. That means if this bacteriophage is specific against Staphylococcus aureus, even any other Staphylococcus will not be attacked by this virus. If this virus is specific against Salmonella or cholera or something else, if this bacteriophage is specific against specific species, it will attack just these species. It will not attack any other bacteria. So, whenever we can find in nature some bacteria nearby, we also can find some bacteriophages. This is how we get a balance in nature between bacteria and viruses. This is how the number of bacteria in nature is kept in balance. And this is how we can treat some bacterial infections. Because bacteriophages are absolutely harmless for humans, for animals, for plants, just because they have their specific target, first of all. And second, I find bacteriophages extremely efficient from more point of views, including economical point of view, because that's enough, let's say just a teaspoon of liquid that contains a little bit of bacteriophages, and you don't need to increase the dose. You put just one, and it will multiply. It will multiply until it will destroy all the bacteria. And when it will destroy all the specific bacteria, it will just leave the body, and that's it. It will not affect any extra cell and any extra flora, any extra bacteria. So it multiplies by itself in the bacterial cell. This is why we don't need to produce too much bacteriophages in order to kill bacteria. Meanwhile, we need to synthesize antibiotics. And if the treatment is 1 week, every day we should apply a necessary dose of antibiotics. And if you have enough, that's good. If not, there is a high chance you will increase antibiotic resistance, and the bacteria will become resistant against these antibiotics. It is not like you put 1 pill of antibiotic and it will multiply just because bacteria is there, like we have in bacteriophages. This is what makes usage of bacteriophages extremely efficient, and moreover, there is no way for bacteriophages or for bacteria to become resistant to bacteriophages. Meanwhile, antibiotics can produce resistance, and bacteriophages will simply penetrate inside and will destroy the bacteria. This is why bacteriophages are largely used in clinical practice, to be frank, not too used. We used to apply bacteriophages long time ago. Frankly, I have no idea why we still prefer more antibiotics. Probably I don't know some details that make some limitations for bacteriophages. But still, it is a very good field for investigations, because it provides a really efficient and harmless way of fighting with bacterial infections. But still, bacteriophages can be applied not just for treatment, but also for let's say disinfection reasons. For example, some sources of water are contaminated with some specific pathogens. That's enough to put a little bit, small amount of bacteriophages in that source of water, and bacteriophages will start multiplying there just because they have substrate for multiplication, that is bacteria. So as a result, we can, without any harm, without any toxicity, we can clean the well or the lake or whatever it is. We can clean it from pathogens. But it will work just in condition that we know which specific bacteria is present there, and we will apply that specific bacteria, those specific bacteriophages for this bacteria. But if we have a lot of different pathogens in there, with a huge contamination with different pathogenic or conditionally pathogenic bacteria, it will be quite complicated to apply this method. And as a result, one of the mostly universal methods of decontamination of water will be application of chlorines and other chemical substances that could be toxic for us if they are in too big amount in this water. This is all we had for today regarding viruses, and we'll continue next week. That's it for today. Thank you.